Last but not least, our final speaker of the day is Dr. Aimée Parker. So Dr. Parker is a research associate in the gut microbes in health and disease lab at Quadram Institute. And her current work investigates the role of the gut microbiota in gut-brain axis in aging. So I'll hand over to Aimée.
Okay, thank you, and good afternoon, everyone, and thank you, Sophie, for the introduction, and thanks a lot to all of the organisers for the opportunity to tell you a little bit about some of our work, and also some of the exciting clinical trials that are happening here at the Quadram. And thank you to everyone listening for staying on to the very last talk of the day. I'll try very hard not to run over. So as Sophie said, I'm a postdoc at the Quadram Institute, and I'm interested in primarily the microbiota-gut-brain axis and the potential roles of the gut microbiota with respect to aging and in modulating potentially neurodegenerative disease development. So today my slides aren't moving. There we go. I'm going to briefly summarize what the microbiota-gut-brain axis is and the various ways in which the bugs in our gut may communicate with the other tissues around the body. And then I'll describe some of our recent work in mouse models where we've used fecal microbiota transplantation to ask how the gut microbes are involved in age-associated inflammation. And then I'm going to jump to talking a bit about two clinical trials we're running at QIB. So the first is the MOTION trial, and this aims to improve our understanding of the links between gut microbial populations and declining health and cognition in older age in a Norfolk cohort. And then finally, I'll talk a little bit about the RESTORE-ME or RESTORE-ME trial. And this is a trial to assess whether microbiota transplantation could be useful to restore microbial balance in ME patients, and whether it can also improve their physical and mental health. So starting then with the gut-brain axis, it's a bit of a trendy area right now, but essentially the microbiota gut-brain axis is sort of a blanket term covering the various types of communication between the microbes in our gut tissues, and the brain. And there are multiple routes and mechanisms by which this type of communication may occur. So firstly, the metabolites and the toxins and the other factors produced by the various microbes in the gut can be detected by a vast array of immune receptors and nutrient transporters and other proteins on the surface of the epithelial cells which line the intestine. And these intestinal epithelial cells, on detecting bacterial products, can in turn release hormones and immune signaling molecules and other factors to act both on other epithelial cells, but also, as we've heard in some of the earlier talks today, on immune cells and also on neural cells in the underlying lamina propria. And some of these factors produced by the cells in the gut can then make their way into the circulation and to other organs, including the blood-brain barrier, for example. And in addition to this, there's a really vast and complex network of neurons in the gut called the enteric nervous system. And this can also communicate information from the gut to the brainstem via the vagus nerve, for example. And then thirdly, some of the microbial products that we've mentioned, metabolites and so on, may translocate the intestinal epithelial barrier, and they can enter the circulation where they then may act directly on other organs. These include things like also neurotransmitters, some cell wall components and other immune stimulators, and also these outer membrane vesicles or OMVs, which researchers at the Quadram and elsewhere have shown can be produced by some particular types of bacteria and can actually cross the gut barrier and disseminate to multiple organs, including the liver and the brain. So there's lots going on, and there's lots of different complex interacting systems, and it's really important to maintain the balance in all these signaling networks. But again, as we've been hearing throughout the day, there's lots of factors that can disrupt these types of signaling networks and interactions. And these include things like infections and illness, particularly antibiotic use and the use of other drugs. Diet and other lifestyle factors are hugely influential, as are our background genetics, and actually even just aging. And in our lab and others, we're particularly interested in the impact of aging, as it's the dominant risk factor associated with a variety of neurodegenerative diseases. And we know that these factors can not just change the composition or the function of our gut microbiota, but actually they can change how our systems respond to some of these microbial signals. So some of the immune system changes that Caroline was talking about this morning. So as I said, we're interested in aging and neurodegeneration, and we know from previous studies that there are some changes in the gut microbiota and actually specific microbiota profiles that have been reported in patients with conditions like Alzheimer's and Parkinson's. And sometimes these changes are referred to as dysbiosis. This is a fairly poorly defined umbrella term really for a change in the diversity or the composition or the function of the microbiota in an unhealthy state as compared to healthy controls. But it's important to remember that there's still really no clear consensus on what a healthy microbiota versus a diseased microbiota actually is in all cases. We also know from large-scale microbial sequencing studies in recent years that the composition and the diversity of the gut microbiota changes with age. And we heard a lovely talk from Lindsay earlier about how the microbiota develops in early life. And we know that this initial early composition then increases in diversity and richness as we go through to adulthood. And actually, in later life, we see a bit of a decrease in diversity and also some altered composition in older age. So alongside these microbial changes, we also have the development in later life of this state of inflammaging. So this is a state of chronic low-level systemic inflammation in which the immune system is primed in a more pro-inflammatory state, and various biological markers around the body and in different tissues show signs of tissue decline and inflammation. In the brain, this type of chronic inflammation is associated with driving damage to neurons and driving the pathology of various neurodegenerative diseases. And one of the big questions we have around the role of the microbiota in all this is whether those changes that you see in older age in microbial function and composition are cause or consequence or correlate of these inflammaging type changes. And this is actually really difficult to address in humans because, as we've been hearing, we're also varied in our genetics and our lifestyle and our drug exposure and our diet and so on. And so early studies, including some of ours to date, have tried to address this using various animal models, including mice. So we used, for example, mice to ask, does the age-associated microbiota influence some of these markers of inflammaging? And to do this, we carried out fecal microbiota transplants or poo transplants. And we used mice from 3 different age groups. So we had some young adult mice, we had some slightly older or middle-aged equivalent mice, and some very aged mice. And we did heterochronic fecal microbiota transfers. That's fecal transfers between different age groups. So for example, young mice received the old or the aged donor microbiota, and vice versa. The aged mice received the very young microbiota. As controls, we also did some coeval transfers. So that's mice receiving their own age microbiota back. So young receiving young, old receiving old, and so on. And we also had some antibiotic treated only controls and some sham transfer controls. And from all of those mice, we collected lots of fecal samples at various time points, so pre and post treatment, and for a couple of weeks thereafter. And at the end of the experiment, we used those fecal samples to do some large-scale DNA analysis of the microbiota, some whole genome shotgun metagenomic sequencing. And this was to analyze the microbial composition and tell us who's there in microbial terms. But we also used that data to do some metabolomics analysis. So not just who's there in a bacterial, a viral, fungal sense, but what are they producing? What metabolites and other products are they making? And I'm not going to go into any of the nitty-gritty detail of the metabolomics and metagenomics today, but just to cut a long story short, we confirmed to start with that the aged mice had a significantly different microbiota to the young mice at the baseline time point. And then we looked in the mice where we'd done the microbiota transfers, have we successfully changed the microbiota? And long story short again is that yes, we can successfully change the microbiota in the gut of an old mouse to look like that of a young mouse, and vice versa. And then at the end of the experiment, we also collected bloods and various tissues and organs, which we used for protein and histological analyses to look at what impact the transfers were having around the body, and particularly we were interested in the brain. But first, I'm going to just show a couple of results from the intestine, so the site of the first interaction between microbes and the host. One of the things we looked at here initially was some hormone-producing cells. So the intestine houses multiple different types of epithelial cells producing a range of different hormones. These include the L cells, which produce glucagon-like peptide 1 or GLP-1, which is a really important hormone in regulating glucose metabolism, and it also regulates appetite. And we also looked at these enterochromaffin cells, which produce serotonin. So these are one of the most numerous hormone-producing cells in the intestine. They actually produce around 95% of the body's total serotonin. And gut-derived serotonin, you may know serotonin is sort of a happy hormone in the brain, but the gut-produced serotonin has multiple roles, including regulating intestinal motility, metabolism, but it can also promote intestinal inflammation. So just taking these cells as one example of many, we found that there at baseline are many more of these serotonin-producing cells in aged mice compared to young mice. But if we took a young mouse and gave them an aged microbiota, we could increase the frequency of these cells in the intestine up to levels comparable to an aged mouse. And vice versa, if we took an aged mouse and gave it a young microbiota transfer, we could reduce the level of these serotonin-producing cells in the intestine. And this potentially has important implications for, like I said, motility and metabolism, but also inflammation. So then we looked at a number of other markers of gut barrier function, including, again, a few which we've heard about earlier today. And these included TNF as a marker of intestinal inflammation. We also looked at serum intestinal fatty acid binding protein, which Neil just gave a nice introduction to in the previous talk. So this is a marker of intestinal barrier leakiness or epithelial barrier damage. And we also looked at serum levels of LPS binding protein, or LBP. This is a protein that's produced by the liver in response to detection of circulating bacterial, like cell wall components in circulation. So it's an indication that bacteria have got out of the gut and into the circulation into places that they shouldn't normally be. So these three markers were all elevated in the intestines and the circulation of aged mice compared to young mice at baseline. And we found that they could be induced or elevated in young mice by transfer with an aged donor microbiota. Conversely, in the aged mice, transfer with a young donor microbiota could reduce these levels of TNF, IFABP, and LBP back down to levels comparable to young mice. So this tells us that the microbiota in aged animals is driving intestinal inflammation, gut barrier leakiness, and it's permitting increased translocation of gut bacteria into the circulation. But the good news is that these effects appear to be reversible in aged mice by transfer with a young microbiota. So moving then, jumping a bit to the brain. And in the brain, we focused on some specific cells called microglial cells. And microglia regulate both immune functions and neural functioning. And when they're activated and so-called skewed towards a pro-inflammatory phenotype, they're associated with driving pathological inflammation and neural damage in a range of neurodegenerative diseases, but particularly in Alzheimer's. So here we used IBA1 as a marker of activated mouse microglia, and we quantified these, shown here in pink, IBA1 positive cells in various regions of the brain. And don't worry too much about the large number of panels here, but these are just representative sagittal sections of the frontal cortex from the mice at baseline and also the mice which were treated with microbiota transfers. So if we look first just at these panels outlined in green, this is the young, the older, and the aged mice just at baseline, and you can see there's an increase in these pink cells or an increase in inflammatory microglia with age. If we then compare the mice receiving the FMT, the transfers pre and post, here we have young mice, and then receipt of an aged microbiota transfer increases the number of these inflammatory microglia cells in the cortex of the brain. And then conversely, again outlined in green, if we take an aged mouse and we give them a young donor microbiota transfer, we can reduce the staining of these cells significantly, so reducing the number or the activation of these inflammatory microglia in a region of the brain that's really important for a lot of higher processing functions. And we did this in a few different brain regions, but I won't go through them all. So just to summarize, we found that microbiota transfer from a different age group, so old to young or young to old, could regulate the activation or number of these important inflammatory cells in both grey and white matter areas of the brain. And so given these results, and staying in the central nervous system for another moment, we also looked at the impacts of the microbiota transfers in the eye, in particular in the retina. And in the retina, we measured levels of a protein called complement C3, which is part of the inflammatory pathway, and also a protein called RPE65, which is a vital part of the visual pigment cycle. So it's involved in the conversion of vitamin A from our diet, and it's really important for proper photoreceptor function and normal vision. And to cut a long story short again, we found that in the aged mice, receipt of a young donor microbiota transfer can reduce the levels of this inflammatory C3 protein, but even more excitingly, it could restore the expression of this RPE65 protein, which is normally progressively lost in later life and with increasing age. So this suggests there might be a potential role for microbial modulation in maintaining eye health and also good visual function in later life. So this is all mice, of course. And just to summarize so far, that if you're an aged mouse or a young mouse given aged microbiota, you have increased intestinal inflammation and permeability, leading to increased systemic inflammation and circulating bacterial products. You have a higher number of activated microglia in some important regions of the brain, elevated inflammation in the retina as well, and decreased expression of this important visual function protein. But the good news is that if you're giving you're a young mouse or you're an aged mouse given a young donor microbiota transfer, these changes are largely reversible back to levels seen in young baseline mice. However, there are, of course, lots of questions remaining, such as is this just a short-term benefit? And how long do these effects last for? So in these experiments, we're only looking at a number of weeks. So is there any rationale for maybe doing a longer-term intervention, perhaps starting in middle age? Is there any impact on cognition or behavior? And we've actually started doing some experiments to look at this fairly recently. And then in the long term, we'd obviously like to know if there's any impact on neurodegeneration, because this is really the important thing for neurodegenerative diseases, and the thing we'd like to try and halt or reverse. And then we would like to know how exactly are the microbes having these effects. And we've heard some nice stories today about particular metabolites or particular immune modulation scenarios. And so we have some hints from our sequencing and metabolic analysis, and also a number of nice papers that have come out in recent months looking at particular species or particular metabolites in aging and immunomodulation. But like I said, that's mice, and actually other researchers have found strong impacts of microbial modulation on health span and even lifespan using fly and fish models. But how does this apply to humans? And so I mentioned earlier that a good number of studies have now shown that the composition and the diversity of the human gut microbiota changes in later life, and that there are these altered profiles reported in some patient cohorts, particularly those with neurodegenerative conditions, as compared to healthy control groups. However, what we don't know is to what extent these altered profiles are contributors to the disease development, or whether they're symptomatic or correlative, and they tend to be taken as one single snapshot in time in already diagnosed patients. So what we now need is a better understanding of the links between the changing gut microbiota in mid to later life and declining cognitive performance, perhaps with other health measures collected over time. And so this brings me to the first trial I want to talk about, which is the MOTION trial. And this is a clinical study being run here at the Quadram. It's in collaboration with the Norfolk and Norwich University Hospital, the University of East Anglia, and it uses the Quadram's on-site clinical research facility and endoscopy facilities. It's a longitudinal study, and its ultimate aim is to obtain some of that mechanistic knowledge of the role of the gut microbiome in normal healthy aging in order to eventually develop strategies that will improve lifelong health and well-being. However, its primary aim is to initially understand the links between specific gut microbial populations and cognitive health and other health measures in old age. So it's going to generate data from serial samples from an initially healthy cohort of around 360 participants who are over 60 years of age. And participants are stratified into 3 groups by cognitive risk, so low, medium, and high risk of developing cognitive decline. There are, of course, a number of exclusion criteria around pre-existing cognitive and bowel conditions. So this includes some dementias, cancers, inflammatory bowel diseases, and so on. Recruitment has already begun, and it's being conducted through several routes. So this includes GP practices, primary care volunteer databases, also the NHS Bowel Cancer Screening Programme, and the local Norfolk and Suffolk NHS Foundation Trust. And as I mentioned, participants will be stratified into 3 cohorts of 120 of low, medium, or high risk of developing cognitive decline. And this stratification is based on some cognitive assessments that include, but they're not limited to, the mini Addenbrook's cognitive exam, or the mini ACE, the Cambridge Behavioural Inventory, the CCI, Cognitive Change Index, along with a whole host of questionnaire-based assessments, in addition to some digital tests, such as the C-Hero Quest and the supermarket memory test apps. The samples, of which there are many, and often, are being collected over 48 months, so they include serial blood and stool samples, some physical measurements and frailty indices, and some less regular but potentially really informative non-invasive scans. So we have MRI brain scans for a subgroup of consenting participants, and also OCT retinal scans for all participants. In a previous multi-center study, I think the participants were between 49 and 69 there, they found that a thinner retinal nerve fiber layer in the OCT scans was predictive of subsequent cognitive decline, even though it was associated with no initially diagnosed neurodegenerative disease. So these could be a really useful screening marker to identify some of the at-risk individuals. Despite the large number of samples required, enrollment is on track and compliance is really high. I think we're now over halfway through in terms of participant numbers. And unfortunately, I'm not actually able to report any specific results as yet because the study is ongoing, but some of the microbial sequencing has begun, so we should, at least fairly soon, be able to start analysing the metagenomic and microbial aspects. So along with these OCT scans and all these other type of questionnaires, stool samples, blood chemistry, and so on, the metagenomic analysis of those stool samples will give us a really good understanding of not just the initial associations and the groupings, but the temporal associations between the changes in the microbiota and changes in any other health measures over the course of the study. And because we're doing deep sequencing of the microbiota, it also allows for potential biomarker discovery. And I didn't mention it yet, but from a subgroup of patients, we're also collecting biopsies, so colonic biopsies, using the endoscopy suite, which we have at Quadram. And this allows us to do several things. So firstly, it allows us to look at those microbes which are really tightly associated with the epithelium and not necessarily really well represented in fecal samples. And we can also use aspects of those colonic biopsies for in vitro models, which we're continuing to develop. And these include the growth and the culture of organoids or mini-guts, and the use of microfluidic organ-on-chip type devices. And these allow us then to test various microbes or various microbial products and really try to get to the roots of their mechanism of action, ultimately aiming then to develop microbial-based therapies based on that knowledge. And I didn't mention, but I should have done this, we're not in that sequencing of the microbiota only concentrating on the bacteria. We are, of course, also looking at the viral and the fungal components, because these could also be really informative. So then to jump tack a little bit in my last few slides, I'm just going to touch on a second clinical trial which we're running at Quadram called Restore-Me or Restore-ME. So this is a phase 2b clinical trial, and it's designed to test the efficacy of fecal microbiota transplantation, similar to what we were doing in the mice, but in ME patients, specifically asking whether FMT in ME patients can restore a balanced or a healthy, whatever that means, microbiome, but more importantly, can we improve measures of their physical and mental health? So you might be wondering what's the involvement of the gut and the gut microbiota in ME. Well, actually, some of the main characteristics of ME include chronic inflammation and dysregulated immune responses, and a lot of those are in the gut. Gastrointestinal symptoms are really very common, and there's evidence of a disrupted or a leaky barrier. So again, some of those markers we were talking about earlier, the LBP, the intestinal fatty acid binding protein, these are also elevated in ME patients. It's already known that ME patients have a different gut microbiota, and this includes a high prevalence of enterovirus infections. And in the bacterial fraction, there's evidence of decreased diversity and an increase in some of those pathogenic or harmful species, and a decrease in what are considered commensal or beneficial bacteria. And these changes, which I'm not going into any detail of really here, are broadly similar to what we see in aging and also some other age-related diseases. So actually a number of studies have analysed the microbiota composition in ME patients in several cohorts from around the globe, and I'm just showing a couple of examples here. But overall, the broad consensus from these sequencing studies is that there's a decrease in one of the main phyla of the gut microbiota of humans. This is the firmicutes. And on the other hand, there's an increase in this other phyla, the Bacteroidetes. And within studies, there's some variation. So while these Lachnospiraceae were found to be the most depleted taxa in this Italian cohort, they weren't found to be changed at all in these American studies. And some of that variation, of course, can be attributable to genetic and environmental variation between cohorts and may also reflect some varying methodologies. But some of these studies do point to specific species as potentially useful predictors or classificators for disease. And like I said, despite the variation, there is this overall consensus that the microbiota of ME patients is different to their healthy counterparts. So this leads to the question of whether restoring a balanced or a healthy microbiota could be beneficial in treating ME. And that's the main question that Restore ME is going to try to address. So it's a single-site, randomized, double-blinded and placebo-controlled trial. It will recruit 160 patients all over the age of 18 with mild to moderate disease severity. Around half the patients will receive a treatment FMT. So this is stool provided from healthy donors, screened and maintained at the Quadram and NHS stool bank. And the other half of participants will receive an autologous FMT or their own microbiota back as a placebo control. And I should mention actually that there is previous evidence that this might be a good way to go. So previous work by Thomas Brody in Sydney demonstrated that treatment of a number of ME patients with bacteriotherapy, but in that case, it was a mixture of selected strains, so 13 anaerobic strains, improved not just sleep quality, fatigue, and lethargy, but also resolved a lot of the intestinal symptoms in a small number of patients. But Restore ME will really be the first trial to formally test the efficacy of FMT from healthy donors and to use some objective measures of improvements in physical activity, cognitive function, and to do microbiome sequencing and blood immune markers and biochemistry. So these are just some of the data collection strategies and also the time points. So the questionnaires and the activity scores and the microbiome sampling and the blood sampling will all be done a week before the FMT and then again at 3 time points thereafter, so 1, 3, and 6 months thereafter. And the primary outcome measures are going to be whether there's an improvement in overall physical activity or any change in cognitive function, including episodic memory or executive function, for example. But there will be some secondary outcome measures as well, and these are primarily around safety and acceptability and tolerability of the FMT procedure in these patients, and also looking at whether there's any improvement in pre-intervention symptoms of gastrointestinal disorders. So the full details of the trial, I don't have time to go over today, but they're in lots more details on the recruitment and the FMT procedure and the outcome measure assessments, all described in a series of videos available on the Quadram website. And the trial is now, after some COVID and regulatory induced delays, poised to start this year. So I think that's almost the end. I'm probably a bit under time, but I was conscious as the last person of the day, I didn't want to run over. So I'd just like to thank everybody involved in the different studies. So in the mouse study in particular, Dr. Stefano Romano, who did a huge amount of work on the metagenomic analysis, which I really didn't talk about at all today, but I'm very happy to if anyone's interested in the bacteria and the metabolomics data. The rest of our colleagues at the Quadram and our collaborators at UCL. The motion study, like I said, involves not just the Quadram, but also the Norfolk and Women's University Hospital and the James Paget. And the ME-CFS clinical trial led by Simon Carding and involving a couple of PhD students within the lab, Rick Hardmans and Catherine Seaton, and again involves partners at the UEA, the Norfolk and Norwich, and with funding from Invest in ME Research. I'd just like to thank you all very much for listening. And as I'm the last speaker, also perhaps on behalf of all the speakers, I can thank Sophie and Rebecca for chairing and moderating today. And I'm just going to leave up some links in case anyone is interested in following up on the mouse data. So we had a preprint out in March last year. The full paper is in press. I don't quite have a link for that yet, but there is a summary video. The motion study, like I said, with the Restore Me study are on the Quadram website if you want to know any more about them. So I'll stop there and say thank you very much.
Thank you for an excellent talk. I really enjoyed it. And we do have a few questions if you have time to answer them. I was just going to start by saying, will you be willing to share some slides because of those links and such with the live audience especially? I'm sure they'd love to see PDFs of your research slides. Would that be all right?
Yeah, sure, I can do that. That's
There's
fine.
no rush. Don't worry, we'll get them to them. So let's go with some questions that we have. So we've got a really interesting one on the YouTube channel from Jasmine, and they'd like to know, would you recommend microbiota screening methods, perhaps as a way of looking at risk for other NCDs or mental issues? I mean, what do you think it could show?
That's a really good question. There is quite a lot of work, particularly from a couple of labs in University College Cork in Ireland, looking at links between microbiota and mental health, not just in a cognitive decline way, but in terms of depression and other mental illnesses and other conditions. I wouldn't say that we're yet at the point where we really could have a screening test or any useful predictors. That's partially what we're hoping to do with the motion trial, is to try and identify some biomarkers or indicators, early markers of later cognitive decline. Some people have described, like many diseases, an altered microbiota profile in people with depression or Alzheimer's, but we don't really know yet what comes first. And because at the moment it tends to be profiling of diagnosed patients, we have no way of knowing whether the microbiota is causative or just as symptomatic or as a result of some other factor. And like I said, and many of the other speakers have highlighted, the trouble with human populations is the variation in all those other factors which are at play. So the diet and the lifestyle, and there's a huge amount to unpick and untangle there. So I wouldn't, to answer the question specifically, I wouldn't yet recommend a screening test for any particular mental illness as the question is phrased. Because I don't think we know enough to do that yet.
But in the long-term future, it might be a possibility.
It may be a possibility. I don't know whether, it depends what the purpose of the test is for, whether you're trying to get an early indication of something which you could then treat, or whether you could have some sort of microbial-based intervention, then that potentially be really useful.
Yeah.
But yeah, we're along in that at the moment.
Yeah. Thank you. That's brilliant. Alison asks on Discord, she would like to know what method you're using for the FMT transfer.
So the FMT transfer, so in the mouse studies...
in humans.
Oh, sorry, in humans. So this is not my area of expertise. I'm very good at doing it in mice. So in humans, we will be doing, it'll be endoscopy delivered. So we have an excellent team of clinical research collaborators and nurses and clinical colleagues, partners with the NHS who work within the Quadram endoscopy unit, and they will be delivering the FMT. And this will be using the stool bank that's an established bank of healthy donors. So this stool goes through a really, really extensive screening program for any potential pathogens or viruses or anything else that may be in there. And there's already a lot of experience within that team in doing FMT for things like Clostridium difficile. So they are really experienced in this, and they know what they're doing, which is good because having, like I said, I'm very much the mouse delivery person, so they don't let me anywhere near endoscopy.
Alison, it's my understanding that a tube goes up your nose and then down your into your tummy and then beyond.
Or rectally. So you can also go the other way.
So for anyone's particularly squeamish, because some people get a bit of a reaction to having something up the nose.
I believe in the FMT trial that I talked about, the Restore Me, that it will be the rectal delivery, but I would have to double check that. I'm not 100% sure. Don't quote me on that. But
I know for the Clostridium difficile work, some of it was done via the nose, but yeah. So Alison, the answer to your question is we're not entirely sure.
Via a tube, via an orifice.
One of two ways.
Yeah. Let's not talk about it too much, really.
Yeah, let's not talk.
I mean, what
about
would
it
be nice, of course, is if ultimately we can get to the point where we don't need to do that, and it doesn't have to be a full microbiota transplant, but trying to move towards those microbial products or specific species that you can deliver in a more palatable way or a less invasive way. So you're taking a capsule, or you're taking a supplement, or you're changing your diet. I mean, this is ultimately the goal. But at the moment, because we have a relatively limited understanding of what we need to change in the microbiota of those patients, this is a fairly kind of blunt, we know it works for C. difficile, can we see some improvement, and then drill down into, can we refine that to a better delivery method in the future?
Joan on Discord is interested to know if there are any potential dangers for FMT, the procedure itself. Are there any risks for the volunteers or patients?
The risk itself is very, very low. Hundreds, if not thousands, of these procedures have now been done for C. difficile patients. And there are very, very, very few isolated adverse effects. I suppose the risk is primarily from unknown things which may be in the microbiota. So although, as I mentioned, these are extensively screened for known pathogens and other things, we don't have a complete understanding of the human gut microbiota yet. So it is possible that you are transferring in something that at some point in the future may have a detrimental effect on health in some way, which we don't yet understand. But the screening team do their very best to screen out everything we are aware of as potentially harmful. But yeah, there is still a risk there, that there is something that we simply don't know about yet.
Got a fairly technical question from Molly now. If you did shotgun sequencing and metabolomics, were you able to identify species and any of their functionally annotated genes which positively correlated with the metabolites you discovered?
Oh, that's a good question. So from the metagenomic analysis, we did identify a number of species which were positively enriched in aged mice when you gave them a young microbiota transfer, for example. So we might suppose that those are potentially beneficial species. They included some of the ones we've already talked about today, so lactobacilli species, also bifidobacteria, some of which we're following up on in vitro work to try and look at how exactly they're interacting with the epithelium and the other organs. And in terms of metabolites, we did some metabolomics analysis based on gene annotation, as Molly asks. And there we found an enrichment in the sort of the beneficial transfers, if we're going to call the young draw beneficial, of specific lipid pathways, so things like palmitate, palmitoleate, and some other long-chain fatty acids. So we've mentioned short-chain fatty acids, and I think Lindsay described nicely how bifidobacteria use the oligomilksaccharides and convert them to short-chain fatty acids. We didn't actually find any change in short-chain fatty acids, but we did find a change in long-chain fatty acid pathways, and also some vitamin metabolism pathways, so some B vitamins, which links up quite nicely with the results we saw in the eye, because we know that some specific B vitamins are really important for maintaining visual health. And so, yeah, we do have several routes to follow there, but we're not yet at the point where we can say this species makes this one particular metabolite that's important. And we are doing a lot of testing to follow those up. So we are testing a number of specific species in a variety of ways to look at how and why they're beneficial. Is it the metabolites? Is it potentially those vesicles that I mentioned, the outer membrane vesicles? Do you need the bacteria alive or dead, like Caroline was saying this morning? And so we're following up on all those things.
I think Aimée and her team are going to be something we should all follow and watch for the next outcomes. Claire asks, do you know of any similar research or trials that focus on the gut microbiota and MS?
Ooh. Claire, to my shame, no, I don't. But that's a really good question, and I will be looking it up immediately after this.
I love an outcome from a workshop.
There's a niggling little thing saying that you passed something on Twitter a week ago. I am sure that people are doing this because of the huge amount of ways in which microbiota can regulate the immune system, and of the interest in trying to find ways of remyelinating or preventing the neurodegeneration that you see in MS, in the same way as we're looking at neurodegeneration as in Alzheimer's and Parkinson's and so on. So there probably is research out there happening with MS, and I can't think of any off the top of my head right now to tell you about, but I will. I'll try and find you in the chat, and if it comes back to me, I'll find you in the chat. Yeah, sorry about that.
Yazim I'm sorry if I've pronounced that wrong, on YouTube says, thank you for the lovely presentation, and asks about serotonin. When it's produced in the gut, does it make you feel happy, or does that only happen in the brain? Any thoughts?
That's an excellent question. So yeah, serotonin we normally think of as being that hormone that makes you feel happy. There are neurons in the brain and other cells in the brain which produce serotonin, and we know that this can go from the brain into the rest of the circulation through the blood-brain barrier. What hasn't been shown, which doesn't mean it doesn't happen, but hasn't been formally demonstrated, is that the serotonin produced in the gut actually has any meaningful transit over the blood-brain barrier into the brain or binds any receptors in the brain. So there are also a huge range of different types of serotonin receptor, and some of those are expressed differentially in the brain or in the intestine. And so the primary functions of the serotonin produced in the intestine tend to be acting on the intestine and other cells locally. So motility, immunomodulation, altering metabolism, also altering neural control in the intestine. So whether gut serotonin makes you happy is kind of maybe more indirectly. It's probably not floating through into your brain and attaching to the right receptors and altering your mood directly, but it may indirectly make you happier by giving you a healthier gut into transit.
Good answer.
But no, it was a great question, really good question.
Okay, we've got maybe 1 minute left, so I'd just like to go back to the questions that you posed on your presentation itself, Aimée. You hinted that in relation to the question, is there any impact on cognition or behavior? And you said you've started to see something. Is that something you can elaborate for us, or is that something we should just follow you to find out more?
No, that's fine. I mean, I'm very happy for you to go to the paper to dig through the supplementary paper, but the figures, but I can just give you an easier answer, which is that in the tests we have done so far, which are some standard tests of short-term memory, and again, this is in mice, we don't really see a significant difference between aged mice that have had an old microbiota transplant, so their own back, or a young one. There's very little difference in performance, and this is actually not very surprising given the short timescales we were looking at. If you really want to change behavior, maybe we should be thinking about much longer term intervention. But a group, again, actually, in Ireland, University College Cork, did some very similar tests, and although they found the same thing as us in terms of short-term memory, they did also do an extra test in which they looked at, it's called the Morris Water Maze. It's basically a measure of long-term memory. Can mice find a platform when they're swimming? And if you train them to do this over the course of the week and then remove the platform and measure the time they spend looking for where it used to be, you get an indicator of whether they remember where the platform was. And this sounds a bit bonkers, but it's a commonly used test in mice. And they claim there that they demonstrate there that they actually show a small improvement in older mice receiving young microbiota in this longer term spatial memory test. So this is in one test, and this is in a longer intervention. So it might just be that in the work we've done so far, we haven't looked over a long enough timescale or probed the right types of memories. There are lots of different types of cognitive tests that test different types of recognition and different types of brain performance in different brain areas. And it's quite hard knowing where to start. So there are hints that it may be beneficial in specific tests at the moment. Yeah.
That's wonderful. Thank you so much for your time.